Really happy to be here today to share some work that is really not just my lab. I'm definitely representing an awful lot of people in what I'll talk about today and point you to the right folks for questions if you have them. So today I'm really excited to tell you about some of the work that we're doing on variant impact and potentially even linking to therapeutics. Something I'm hoping we're all excited about our research ending up in. So first of all, this is where a lot of us are really focused, that we care about the fact that there are many, many more sequencings, many, many more variants in our databases these days, and they all need characterizing. And so, of course, it's really awesome if you can make some kind of an assay to analyze, for a single gene, analyze tons of variants at once, and we've seen some awesome examples of that. It's even better if we can do lots of genes and lots of functional readouts at the same time, and that's what I'll be talking about today, is different kinds of ways to profile a variant. In other words, measure lots of potential readouts for a given variant. And what you can do when you do this is not just gain phenotypic insights about each gene's function or each variant's impact on that function. Those are the phenotypic insights, but also you now start to have patterns where you can group classes of variants and figure them out. So instead of having 157 variants in your gene of interest, you may end up with, okay, well, actually there's only 3 classes, and that's a reasonable thing that I might be able to study. And so we'll talk today about a couple of ways where these kinds of insights can hopefully lead to drugs for treating the diseases that we all care about. So the first project or actually I guess most of the talk is structured around this project VarChamp, where we are aiming to characterize the Mendelian proteome. It's an awesome group of people. I'm guessing many of you know many of the people on this slide, not just the big pictures, but also all the folks that are involved, many of whom have had posters here on different aspects of this project. But it's a really cool project that is made possible because of NHGRI's IGVF project. Consortium. And so all the work, I'm very sorry I couldn't figure out how to figure out who to put on which slide for all the different kinds of results. So suffice it to say that what I'm talking about is work done by this whole collection of people individually. So first step, if we want to characterize a lot of variants, is to clone a decent collection of variants. And so for this, we're aiming to actually capture 80,000 coding variants across thousands of genes. And you can see where they're coming from here. Thanks to many of you in this room and other collaborating consortia, we have some nice overlap with other projects that are out there in the world, so that we'll really have a lot of data to cross-compare eventually. If you'd like your gene or your variants to enter this project, it's not too late. You just go to the MAVE registry, MAVEDB, and put your information in there to suggest that it be prioritized. And I mean, just being in there will suggest that it should be prioritized for this project. So you might ask, how exactly are we selecting these genes? And the answer is an awful lot of Zoom calls and discussion, but fundamentally our iterative process is first to, it's just really nice that we have the timeline here to be able to first benchmark genes using pathogenic and benign variants, make sure that we can see in at least one of our assays, can we see some kind of differential between pathogenics and benigns in general? And if yes, then we can go off and clone some more VUSs. And so the goal, we also want to cap it. We don't want to do a ton of work on a single gene, but instead spread it out across a lot of the genes that are in the database. So those are the variant collection that we're interested in. Oh, and I should say, everything I'm showing today is sort of public information. You're welcome to take a picture, but I've also uploaded slides, and I'll show you the URL for that at the end of the talk if you just want to get a PDF of it as well. Okay, so now once we've cloned a collection, I should say it's an overexpression collection. So we're overexpressing the protein with and without a given variant that's in ClinVar or wherever. So the second step is figure out how to capture the impact of that variant. And if you care about a single gene, then the steps here would be figure out the function of that gene, make an assay for that gene's function, and then carry out the assay for all of your variants. Here, we want something that's widespread and applicable across all the genes and all the variants. What kinds of assays can we run that give us the greatest opportunity to identify some kind of impact of a variant across all kinds of gene functions? And so the collection of assays that we're running are shown here. And I'm going to go through each of these systematically. So let's start with the first assay is based on protein-protein interactions. So here, this is called edgetyping, and it's systematic mapping of the protein-protein interaction profiles. And again, not my lab's work. This is the rest of the consortium that's working on this. But it's a semi-quantitative yeast-2 hybrid that measures these protein-protein interactions in the presence or absence of a given variant. And so we're overexpressing the protein. We're seeing what are the top proteins that it normally interacts with the reference version of the protein, and then making a bunch of different variants, as you can see here, and measuring how that changes the impact. So that gives you a basic view of how that looks. And what we can also do then with those kinds of profiles is we can start to group variants with each other to see if they fall into certain classes. In this case, it's largely the variants that mess up all the interactions and the variants that don't mess up all the interactions. But there's some nuance as well that allows us to take a look at how impactful does it seem each mutation is being in terms of interacting with the rest of the proteome. So that's the first assay. The second assay is an abundance stability assay. This one's in mammalian cells. And here it relies on a reporter GFP attached so that we're measuring the fluorescence of GFP relative to a transfection control. And here, this assay is a pretty nice one to run using flow cytometry, and we've ended up characterizing so far a few thousand variants, but the first 1,000 are shown here, and you get a sense of, I'm pretty sure that's not a mistake, that you can't see the benign bar chart there, but it really has a dramatic difference between pathogenic and benign. And then what we can do, since we have a decent amount of data for assay 1 and 2 so far in this project, we can look at the correlation between the interactions and the abundance. So we have interactions on the left that I showed you, protein abundance is on the right, and here you can see a pretty clear picture that when there is less and less of this protein expressed, these interactions start going away, and so it's maybe not too surprising of an interaction. All right, so those are those 2 assays, which I know much less about, and I don't know if Mike Calderwood is here. Yep, he's back there, so he'll answer any questions you might have about that sort of data. But now we'll turn to my specialty, which is image-based phenotypic readouts. So we'll talk about 2 of them today, localization and morphology. So I have to take a little sidetrack here for those who are not so familiar with images as data. So let's talk about how do we get phenotypes out of images. So what we do, we're typically working in high throughput, lots of samples, and so that means we're working with automated microscopes, and it means we're working with automated image analysis. So we do this microscopy, we capture all these images, however many channels we may have, and then we run some kind of software to segment the cells in order to identify the different compartments, like nucleus and cytoplasm, of the cells. And that's pretty straightforward for the mammalian cell type that we're using in this project. In this case, it's U2OS cells. It's an osteosarcoma cancer cell line. So segmentation is not typically too hard. And then the next thing we do is we extract all the things. We extract all the features we possibly can. And this includes, nothing here is overly complex or fancy. I mean, if I explain every feature, there's thousands of features that come out of the software, but I really can't explain every one of them to you. Some of them are based on counting, like how many speckles or how many cells are there. Shapes and sizes are self-explanatory, but remember, we're looking at different compartments of the cell, not just the overall shape of the cell. We're also looking at intensities and textures of whatever channels we have present in the experiment, so that gives us sort of smoothness of different stains and so on. So all kinds of things that we can measure, and it adds up to thousands and thousands of features because it's quite combinatorial depending on how many different stains we have, how many different compartments we have, how many different features we have, and each of these categories has dozens to 100 or so features in it. All right, so that's how we turn images into a numerical profile that represents kind of a transcriptomic profile or a proteomic profile. It's the same shape and size of data, and of course, it's inherently single cell. And I should also say it's exceptionally cheap compared to transcriptomics. It's on the order of maybe 10x cheaper, and proteomics, it's probably 100x cheaper. So when we're using images in this way, you can kind of see that instead of just looking at each set of images and saying how much GFP is there, or do the cells look small or large, instead, which would be kind of like a screening approach, instead we're taking advantage of all the data that we have present in the image in order to read out lots and lots of phenotypes at once. So here's a big view of that in case you tuned out and want kind of the big picture view. Image-based profiling is where we take some kind of perturbation, in this case, reference and then variant number 1, variant number 2, and so on. Take all those different perturbations, treat the cells, image the cells to see what their response is, and then turn those images into this numerical matrix, and then we compare those matrices just as if they were any other kind of profile. That allows us to look for similarities and connections for all kinds of purposes, but today we'll mainly focus on variant characterization. I'll just show one example from outside the world of variant characterization where we've used this approach to group chemical compounds based on how they function, like what proteins are they targeting, what mechanisms in the cell are they disrupting. And I show this one because it gives us a nice comparison of mRNA profiling and image-based profiling. And if I asked, I should have asked before I showed the data, but I would have expected that mRNA profiles would be more powerful than just a single image-based assay. It seems kind of intuitive that certainly measuring, in this case it's only 1,000 mRNAs because it's the L1000 mRNA profiling method that is feasible to run in high throughput, but still measuring 1,000 mRNAs should be better than snapping one image of cells. But it turns out those two data types are actually pretty comparable in terms of their information content, and even better, they're fairly complementary, that there's some mechanisms of compounds you would see using either type of profile, but some are unique to just mRNA and some are unique to just images. So we think it's a really nice data type, especially given the cost comparison. All right, so now we're back to the VarChamp project where we can look at how are we measuring localization and morphology. So now we turn to a collaboration that, with Mikko Taipale's group at University of Toronto. So he's generating the data for this aspect of the project, and we'll start talking about a particular subset of the data that we've analyzed so far, where we are taking 3,500 disease variants, and his student Jessica Lacoste, she tagged and overexpressed and imaged that large number of variants together with their reference counterparts. The tagging is in the green channel here. We also have a few other channels as landmarks in this experiment. And our mission is to see if we can identify, does having this variant present versus the reference sequence, does it cause any change in the protein localization or the morphology of the cell? And excitingly, in this experiment, we identified a couple hundred phenotypes, and I'll show them here. Actually, I'll show you one of them here. This is a gene, LZTR1. I don't personally know what it does, and what I do know is that there's a mutation in it which is known to be causative for Noonan syndrome. And while I personally couldn't look at a child with Noonan syndrome and diagnose them, they look pretty much like any other kid I've seen, the computer is able to look at images much more impressively than I can, but you don't even really need a fancy computer to tell you that I don't know what this protein is, I don't know what it does, but it looks different. The localization of this protein is just different when that variant is present, and we don't know for sure that that localization difference is what is causing the eventual phenotype that the child experiences, but it's not a bad place to start. And so if we can identify these kinds of phenotypes that are associated with pathogenic variants, then we have the potential to test drugs to reverse the phenotype, which I think is a really, really exciting aspect of this. All right, so then this big image has to load, which shows you us meaning Mikko and Jessica, scaling this up, and then Marzia helped them with the computational analysis to just identify tons of these different pairs. When this variant is present, you see a change in localization. It's 250 variants out of the 3,500, so as far as a percentage goes, it's not incredibly high. Between 5 and 10% of the proteins have some kind of localization phenotype, but for a single assay that you can run across lots of diseases that is pretty cheap, it's a pretty good yield, I would say. And we can also look at things like what kinds of localizations are more common. We can also imagine things, well, if somebody comes up with a drug that works for one of these disorders that causes a localization change from point A to point B, maybe all the other diseases in that category might be well treated with that same drug. So it offers some kind of therapeutic hypothesis, at least in the absence of any other information. Again, it's a good place to start thinking about what might be useful for a given disorder. Here's a little picture that gives you a sense of the spread of the variants that were tested in this experiment, out of showing you what % of the variants are mislocalized. And pathogenic, 15% of them are mislocalized, whereas for the other categories, it's much lesser, closer to 5%. And again, now this is the third assay that we're scaling up right now and testing all 80,000 variants. Okay, and not as part of the whole VarChamp variant characterization project, but as a side project launched with JT Neal and Paul Blaney here at the Broad, we thought, okay, 250 disease phenotypes, 5 to 10% of the total variants that exist out there, but that's a heck of a lot of disease phenotypes where we could take each one and go do a drug screen and try to find compounds that can reverse the phenotype, as I said. That would be amazing if we could do it, but we can't afford to do even a dozen of these kinds of experiments, and so of these large-scale drug screens. And so what we would like to do is that we would love to take the 6,000 drugs that have been treated in humans. Either they're a currently marketed drug or maybe something that's been pulled from the market, but at least it has been in humans, and we have known toxicity and safety data on it. Can we take those 6,000 drugs and figure out a way to test them against the couple hundred different disease phenotypes that we found so far? How on earth are we going to do that? And the answer is making use of this barcoding strategy, pooled optical profiling, for those who have heard of it. And basic idea is this. First, we capture, in each well, we put a single compound, and we're asking the question, does this drug cure any of these diseases? And so does it cause any of the disease variant protein localizations to revert back to what we would expect the healthy reference protein to look like? And so the first step is to capture the morphology and the localization of those different proteins. And then the second step is to wash those stains away and then carry out this in situ barcoding that tells us this little island of cells got this particular gene overexpressed, and it was the reference version. And this other island had the variant of interest. And by comparing those and their responses to each drug, we hope to be able to scale up to the point where we can do a few hundred drug screens in parallel in a reasonable amount of time and money. So this project is just getting underway, and we're just working on pilot experiments for it. But we're pretty excited about the potential downstream impact. So thanks to all of you who have discovered the variants that ended up in the catalog, that ended up being cloned. I mean, there's many people in this community that have taken many of the steps that have gotten us all the way to the point where we really have the potential to find compounds that are promising for these different disorders. All right, now back to the main thread. The last assay is focused more on morphology. So let's take a look at that. Here I will again turn to pooled optical profiling. Here again, it was a collaboration with JT Neal and Paul Blaney's group, as well as Calico, that funded us to create this really lovely data set that's now public, where we are knocking down each gene in the genome using CRISPR in a pooled format. We have three genomes worth of data that's out there in 2 different cell types now. And the output here is an image-based assay. I won't describe the details of it, but called cell painting, where we label the 5 channels that are available on the microscope. We use to label all the different organelles in the cell. And so with that readout, we, again, capture all that numerical quantitative profile out of each individual cell, and we use the barcoding to figure out which cell got which reagent, so it can be done in a pooled format. And so then that allows us to cluster all the genes in the genome based on their morphological profile. So which genes cause the cells to change morphology in different distinct ways. Some of them quite subtle, some of them quite dramatic. And this shows, the plot itself just shows you the genes that have a phenotype that are different from negative controls. And this paper just came out as a preprint last year and has just some lovely tidbits in it. I recommend taking a look. Okay, so let's see. So what does this have to do with variant functions? Well, since this kind of thing works, we can now characterize alleles. So that was a CRISPR knockdown, where we're looking at loss of function, and that may very well be useful to many of you wanting to understand the function of a given gene. If you find that your given gene is in a cluster with a bunch of genes that have a known function, that gives you some hypothesis to follow. But here, the second experiment I'll show you is based on overexpression. So now we are, again, overexpressing a protein that either has a variant in it or has the reference version of a protein. And it's a really cool kind of experiment. Again, we're imaging the cells, extracting these numerical profiles, and then clustering them. In this case, the cluster are across the diagonal, and you can see the reference version of BRAF in this case is down here. It actually clusters with negative controls. So in other words, overexpressing this protein, the cells don't really care about. They don't really see anything. But when you overexpress many of these other variants, you see a dramatically different kind of phenotype. And they cluster in different groups, and those groups, turns out, from what is known, are meaningful, such as the top 3 variants are known to be not constitutively activating, whereas the rest are all kind of similar. So you can imagine that if there were a drug that's known to be effective for one of these variants within this pathway, if you've got a variant of unknown significance and it clusters nearby, it at least gives you some sense that maybe your variant has the same impact as another variant that's been better characterized. So we really love this approach and have scaled it up. In this case, it's only a few hundred genes that were associated with lung cancer, but it gave us confidence to use this assay for the much larger VarChamp project that's just getting underway. So I don't have any data to show from the latest batches of this, but this gives you at least the proof of concept of what we're aiming to do and the kind of data that will be available to the community once we get this really up and running. We just have like the first few thousand alleles that have just come through the pipeline so far, so this will be coming out soon. Just a little side note for those of you who are excited about single cell. It's really fun to look at this kind of data in a single cell way. So this is a, I don't remember if it's a UMAP or some kind of embedding of the morphological space, and each point is an individual cell. So we can look at, in this case, the red wild type cells occupy a certain portion of space. In this case, a particular mutant is in blue, and it looks pretty much just like the wild type version. So the Venn diagrams overlap pretty much, and we would say, yeah, this mutation doesn't seem to offer much of an impact on gene function. By contrast, if we look at a different gene, we see a different pattern where, again, in this case, the wild type reference version of the protein is quite, has some distinction from control cells, but then they get even more extreme when you look at the particular mutation that's being examined here. So now we see a much higher variant impact. So we'll be able to provide all this kind of data for the data that we're producing for VarChamp. Okay, so how do we turn this into drugs is my last little chunk. And the answer is shown here. And it relies on the fact that we have some really nice public databases of this kind of imaging data, again, using the cell painting assay, which I haven't really emphasized, but is what we're using. So let's say we have a particular gene that we care about, and we either overexpress it or knock it down, or overexpress it with and without a variant in place. The cells look a certain way. Hopefully they look a certain way that's different from control cells. And if that's true, even if it's just the computer that can see it and not our eyeballs, that allows us to match it against a whole bunch of drug profiles that are out there in the public. This shows 30,000 that we had at the time of doing this experiment, but we now have more than 150,000 thanks to a very big pharma consortium organized. And with more than 12 companies involved. So that was a really beautiful resource that allows us to do this. And what's really wonderful about it is that it massively reduces the candidate compounds that might be useful for impinging on your particular gene's function. And so in this case, we ran an experiment where we already knew what which compounds should work for which genes, and found a nice success rate. That's not shown here. But then we did an even harder thing, which is go looking for compounds that we didn't know existed and would impinge on a certain gene. And we found a pretty decent success rate finding compounds that work. Keep in mind, this is a step that pharma companies will typically spend on the order of $10 million over the course of a year, creating an assay to interrogate a function and then screening 1 million compounds in order to find a handful of good ones that work. Here we can do it just by this computational query, and it takes an afternoon to do. So one in particular that we found was a regulator for a compound that could impinge on the function of YAP1. And in particular, they kill sarcoma cells, and that caused us to found a company aiming to take this general strategy and scale it up for solid tumors especially. Okay, so that's what I have for today. Many more things you can do with image-based profiling, treating images as data in this profiling kind of way. Just a sample of some things are shown here. You can see the URL for getting the slides is shown here that you're welcome to download if you'd like. And shown there again, thank you so much for your attention. Yeah.
Have some like success stories that could highlight?
So we don't have a single attempt to do this with variants specifically. So far the only case where we've done it is overexpressing a gene, which in this case is maybe producing a dominant negative, so essentially a loss of function phenotype. So the only cases we've done it is using overexpression. So we haven't tried it. In theory, it seems like we should be able to make it work, so we don't have a sense of the percentages. But at least for that overexpression case, I'm sorry, I can't remember the percentage from that paper, but I feel like it was on the order of 42% of known gene compound connections were found in the experiment, so that's not too shabby.
Oh, no, yeah, that makes a lot of sense.
Yeah.
It's really, the question, of course, is like how many unknown ones are there?
Yeah,
right?
yeah.
And that's known, but
yeah. So hopefully on the order of 42% of the time we'd be able to find some unknown some new compounds for genes that we're excited about.
Super cool.
Yeah.
Hi, Bill Mowry, Alexion Pharmaceuticals. I had a sort of a question along the same line. So in matching the profiles for drugs to known overexpression or loss of function, you can imagine if you're targeting the gene product perfectly, you would see like an exactly opposite profile. But are there cases of like drugs that are dirty or have like off-target effects where you can sort of see that the direction isn't directly opposite of what you see when you overexpress, but it's sort of like has off-axis kind of effects?
Yeah, so there's two elements of that. One is polypharmacology, like a lot of drugs are known to hit multiple targets, and so then what is that profile going to look like, and is there any hope of deconvoluting it? And I feel like that's a really interesting question that has not been addressed too much in the field so far. There is one paper from Greg Way that attempts to address, like computationally, what would it look like if we perturb gene A, we perturb gene B, what would perturbing both of them look like? And so he's made an attempt at that, kind of deconvoluting the profiles, but I think it's a really challenging area. The other question is the directionality, and here I just have to be generally confused because we often see the directionality as sort of the opposite we would expect. I mean, if you were just taking a very straightforward approach to this, you would say, well, if I knock down a gene and I have a compound that inhibits that protein, that should be a match, whereas maybe if I overexpress a gene, I should get the opposite direction. Or even just looking at if I overexpress a gene or I knock down a gene, those should be opposite. And we find every possible combination of things going maybe the way you would expect versus the way you wouldn't expect. I mean, there's obvious reasons why. Maybe it's a dominant negative. Overexpression might be a dominant negative. That explains why it would match a knockdown, and all kinds of compounds do all kinds of weird stuff to proteins and cells as well. But we've gotten to the point where I just almost don't even care about directionality anymore. If the profile either matches or anti-matches our protein of interest, we'll pursue it just to see what happens. Worst case scenario is you've got a tool compound that like makes the disease worse or like mimics the disease in a dish, and at best you've got something that's actually helpful.
Phenomenal talk, thank you. Anna Gloin from Stanford. I'm fascinated by the decision for the cell type that you operate in. And I wondered if you had evaluated different cell types and what the sort of spectrum of effects is and why you landed on your, I think it was an osteosarcoma cell line as your go-to cell?
Yeah, so for people who do a lot of microscopy and care about cell biology, you'll probably recognize U2OS as being just a very nice flat cell type. So I'm sorry that we don't have any more justifiable reason for having used, we didn't know it was going to be so powerful and exciting and interesting when we did the first little experiments, but the first ones were done in U2OS, and then we said, oh, well, we've already got data made in U2OS, so we may as well make more. So most of the public data now for cell painting is in the U2OS cell type, but we have done some comparisons. If you pick some other cancer cell line, you certainly don't get the exact same answers, but I would say you get largely similar answers. So just ballpark, like 80 to 90% of the gene-gene connections or compound-gene connections, like maybe something on that order is kind of going to be the same across a reasonable set of cancer cell lines. But I think what's really exciting is using like primary cells, and so people have done neuronal cell painting and cardiomyocytes and adipocytes and all kinds of lovely flavors using primary cell types. And I think it's a really exciting direction to go. So for the VarChamp project, we're just using one single cancer cell line right now. It certainly makes the experimentation a lot more practical. If we were to try to do that in primary neurons, it would be much more challenging, although there is a site, another site of the IGVF consortium that is doing some fancy cell lines with image-based readouts. So it's just, the scale has to be much smaller because they're much more finicky to work with.
I think that's all the time we have questions for. Thank you so much.